Hello students, I'm going to lecture on the endocrine system and the controls of the endocrine system, and I will also talk about some of the pathophysiology of the endocrine system in this lecture. The chapter readings that you are seeing on the slide correlate with Guyton and Hall 12th edition, but this lecture may likely be assigned to both my anatomy students and physiology students and pathophysiology students. It's weird to think that everything that's shown here on this picture is an endocrine gland. That's right, everything. Even the adipose tissue that you see here, even the stomach, even the kidney. I'm going to lecture on many of the glands that you see in this picture. Most notably, I'm going to lecture on the basic review of hormone actions, the classification of hormones, and I'll tell you about how hormones are released. Is it neural, humoral, or hormonal? Then I will focus on the hormonal pathways, and this will entail a discussion on the HPT axis, and I'll talk about the anatomy of the thyroid gland, the actions of the thyroid gland, how we make thyroid hormone, clinical issues with the thyroid gland, hyper and hypothyroidism. This applies to the pathophysiology. And we'll discuss how a patient can have hyper or hypothyroidism. Then I'll move on to the adrenal glands and talk about the HPA pathway, what it's supposed to do, how it's supposed to act, the normal physiology, and then of course I will highlight some of the pathophysiology of the HPA pathway. I apologize, I'm a little mucusy today because of allergies. Growth hormone and parathyroid hormone and calcium regulation that goes along with parathyroid hormone will cap off this lecture. You should know that a hormone is a type of chemical signal. And when we talk about the endocrine system, in particular hormones, we're talking about cell signaling. That's really what we're discussing. And the word hormone and its definition has morphed through the years in the literature. And I'm going to be very pure in my approach to its definition. A hormone is a chemical signal that is distributed into the blood. It is released by a cell or gland without using a duct, so it's through ductless means, and into the bloodstream, and can have a target cell that is very distant away. And the target cell is a target cell because it has a receptor, a functional receptor that can bind to that hormone, as shown in this picture. You can see cells releasing their hormone, and the hormone is distributed into the bloodstream, and target cells are only target cells if they have a receptor, functional receptor for that hormone. Hormones are very different than other types of chemical signals shown here in the picture as well, where you see one cell releasing a chemical signal to an adjacent neighboring cell. That's called paracrine, para meaning next to. And hormones are very different than what you're seeing in the bottom of this picture with autocrine, where cells are releasing hormones that chemical signals that travel very short distance and bind to receptors that are on the very same cell. So we're going to be very pure in our approach that a hormone is a chemical signal. It's part of cell signaling, but it is a chemical signal that's released into the bloodstream. Now, what can bind the hormones that are released? You're seeing here a cell that can respond to two different hormones in this picture. How a cell will respond is based on its pre-programmed response, and by that I mean its gene expression. And for those of you who know me, those are two of my favorite words. The ultimate goal is to change the physiology of the target cell. How can this be done? It can be done by changing the activity of the current proteins that are already expressed and functional inside that target cell. Protein activity can be changed most notably by removing or adding a phosphate group to a protein. But we also can change cell physiology by changing gene expression of that downstream cell. But all in all, whether it's phosphorylating or removing a phosphate group from a protein or increasing or decreasing a kind of protein that's being made, overall, all of that means that the downstream target cell is going to change in its physiology. I have a question on the screen. What would happen if there was a defect in the gene that codes for a protein hormone? Or what if there was a defect in the gene that codes for the receptor for that hormone? Or what if there was a defect in the cell's machinery to even make the hormone? Well, then it would be as if the signal was lost altogether. If the signal isn't made, if it's not made appropriately, or you can't bind to the signal, any of those ways, it's as if the signal was never released at all. Hormones can interact with each other. We can see redundancy. We can see antagonistic responses. Some hormones are even considered to be, quote, permissive to other hormones. When hormones work in redundancy, when they augment each other, we say there's synergism. And that's shown on the screen, where you can see blood glucose levels most definitely are affected singularly by cortisol, glucagon, epinephrine. But notice how blood glucose levels change when we add 2 and 2 together, or if we have all 3 working synergistically. Notice this top curve of the graph where we have glucagon, epinephrine, and cortisol working together. If we were just to add the 3 responses of the 3 hormones individually added together, we might predict something going in between the curves of glucagon plus epinephrine versus glucagon plus epinephrine plus cortisol if we added these together. Clearly, when all 3 are working at the same time, there is a greater response with the blood glucose levels rising very, very well. That suggests to a physiologist that the combined effects have much greater results than what you would predict by adding each of these hormones and their effects up in a singular fashion. That suggests synergism. Antagonistic hormones means they have opposing effects. An example of that would be growth hormone versus insulin. Growth hormone promotes more blood glucose levels. Insulin opposes that. And yet the 2 of them are released at the same time to ensure proper growth. And that's a fun story to talk about too. Most students think insulin is just for glucose to be moved into the cell, and that is not its only case. Insulin is used so much by cells of the body, and it's not just about glucose transport. It's about amino acid transport. It's about activity level, the sodium, potassium, ATPase. More on these stories to come. But insulin is released when growth hormone is released, most importantly, not so much for antagonistic membrane transport of glucose, but to ensure that that adequate amounts of amino acids are internalized into the cell. And growth hormone needs those amino acids to go into the cell to ensure more protein synthesis or anabolic pathways of protein. Another example of antagonistic actions would be between estrogen stimulating a receptor, say, on a cancer cell in the breast, maybe a cancer cell that feeds off of estrogen. And we can administer tamoxifen, which is a drug, And it binds to the same receptor, and this means we can use antagonistic actions even in a drug aspect so that cancer cells don't get proper fuel, meaning they don't get driven into a more mitotic rate because they're getting stimulated by estrogen. We can compete for it. This is called competitive inhibition. Permissive hormone, as you see on the screen, really applies to thyroid hormone, is a perfect example. If a woman comes into your clinic and is complaining of excessive weight gain, her menstrual cycle is off, if her husband says she's not being logical in her thinking, one of the first things that a physician is going to rule out is actually a thyroid hormone imbalance. Because in order for any other hormone in the body to work appropriately, appropriate levels of thyroid hormone have to be around. That's what we mean by permissive. Thyroid hormone levels, a properly functioned thyroid gland ensures that the other endocrine glands and their hormones will have the most optimal effects. What is a receptor? A receptor is a protein, and proteins are expressed from genes. We've talked about gene expression. And a protein being made from gene expression means that if you're going to have a functional receptor, you best have a gene that is not mutated nor damaged. Where can you find a receptor for a hormone? You can find it in the cytoplasm. You can find it embedded in the plasma membrane, or you can even find the receptor in the nucleus too. But receptors have a certain, proteins are folded, and when a receptor is folded, when a protein that is acting as a receptor is folded, there's a binding site, and it's like a perfectly fitted glove. That's why I have the glove up here. And the hormone, the chemical signal, can only fit in that receptor. It can't just aberrantly bind to other receptors, at least in an ideal world. And the idea is that once the hormone binds to the receptor, then that causes or instigates a change in the physiology in that target cell. And what kind of change is it? Well, that depends on the gene expression of that cell. I've already answered these questions. Given that a receptor is a product of gene expression, if there's a mutation, you have the opportunity of losing the functionality of that receptor. If it was denatured, you lose the functionality of that receptor. And again, it would be as if the signal was never released. The cell would not change its physiology. Let's talk about the fates of a hormone. What can happen to a hormone once it's released into the blood? Well, some hormones need a blood carrier. So they're carrier bound. Some hormones can actually reach their destination and bind to their receptor and cause changes in the physiology, the biological effects. But in order to bind to the receptor, the carrier would have to release them. So that's why you see in this picture the reversible arrows for a carrier-bound hormone. Some hormones don't make it to their receptors, and they're degraded either by enzymes in the blood or even within the cell, so they don't make it to their receptor. And some hormones actually need to be activated. They need to be converted into an active form. Some hormones might not make it to the receptor because they're removed from the body, say from the kidney, or they might be degraded by enzymes that are found in the liver. Either way, some hormones don't make it to their receptors. What are the flavors, as I like to say, of hormones? Hormones come in a basic 3 classes. They're either steroids, which means lipid-derived, or they're long chains of amino acids, and those are peptides/proteins, or they're derived from a single amino acid. And an example of that would be tyrosine, and how it can be converted into dopamine, norepinephrine, epinephrine, and even used to make proper thyroid hormone. In no way do I want my students to memorize this picture. It just shows you that the largest class of hormones is the protein/peptide class. The smallest hormone classification are the single amino acid derived hormones, the catecholamines, where they're derivatives of tyrosine, as shown here. And tryptophan is shown on the screen as well. Tryptophan can be used to make melatonin, and tryptophan can also be used to make tyrosine, which can be converted into epinephrine, norepinephrine, dopamine, and even used for thyroid hormone. More on that later. But again, the largest category are the peptide-derived, either full-blown large proteins or smaller peptides. Let's talk about steroid hormones. They are synthesized from a precursor, cholesterol. And scientists and chemists, whenever we see a whole bunch of rings together like this, it immediately tells us a lot about the behavior of that compound. And that tells us there might be some hydrophobicity, not water-loving, lipophilic. So if you're lipid-derived, you love lipids. Like loves like, you might have learned in chemistry. If you are lipid-derived hormone, you don't like being in the watery plasma. Lipid hormones, steroid hormones, need to have a carrier. And because they have a carrier, a protein that envelops around them and protects them and basically affords them more water solubility, these hormones tend to have a longer half-life. They last longer in the body. They're not as readily filtered and cleared from the body in the form of urine or destroyed by liver enzymes or blood enzymes, so they last longer. And because they are lipid-derived, they can also cross the cell membrane, and their receptor is most likely found in the cytoplasm or in the nucleus. So we say they can go all the way. They can cross the cell membrane. And these steroids are most certainly going to target changes in gene expression very, very strongly. The fastest acting steroid hormone can make physiological changes in a target cell in as little as 45 minutes. And that's pretty remarkable to me. Steroid hormones, like I said, have their receptors in the cytoplasm or even in the nucleus, but both of these cases certainly is going to change expression of the genes. We're going to get new proteins being made and other proteins being shut down, not expressed. Either way, the physiology of the cell changes. Thyroid hormone is a quirky hormone. On the previous couple slides, you saw that thyroid hormone is derived actually from tyrosine amino acid residues. And yet, it has the action or behaves as if it's a steroid hormone, even though it's not. And the reason that it behaves like a steroid hormone is because of its chemistry. It has this benzyl ring. And to make things even more interesting, when we make a thyroid hormone proper, we actually take two tyrosine residues and link them together. So it almost looks like you have half of a cholesterol molecule. Thyroid hormone, even though amino acid derived, is not very water-loving. It will need a carrier in the plasma. It will have a longer lasting half-life. And we say it most certainly goes all the way. It's going to bind to its receptor in the nucleus. So thyroid hormone, I like to say, is a hormone that is a wannabe. It's amino acid derived, but it wants to be like a steroid. Protein or Peptide and amine or amino acid based hormones share some common characteristics. They tend to be water loving, hydrophilic, and that means they're better able to be released into the plasma, less likely to have a carrier protein. However, this comes at a price. They can't get across the cell membrane, and the receptor is traditionally on the surface of the cell membrane, so they have an integral membrane receptor. The notable exception to these categories, of course, like I just mentioned, is thyroid hormone. Although amino acid derived, and you would predict that it's water loving, it really, because we have two tyrosine residues linked together, it behaves more like it's a steroid. More on thyroid hormone later in this lecture. Then we have the synthesis of the amine hormones, this summary slide. Again, typically water loving, don't need a carrier receptor on the cell membrane. And this is showing you how tyrosine can be used to make your catecholamines. And yes, dopamine, even though used as a neurotransmitter, is also used as a hormone in the body. Norepinephrine is mostly used as a neurotransmitter in the body, but some is released into the blood as a hormone. Most norepinephrine is converted into epinephrine, which is highly used as a hormone in the body, though there are areas where it's used as a neurotransmitter. But they're all tyrosine derived, and you just make chemical modifications with each step using enzymes. Do not memorize these enzymes. I'm just showing you the pathway. But notice that tyrosine is also used to make thyroid hormones. Yes, there's more than one flavor. And I have a separate arrow for it because I want you to remember that even though it's amino acid derived thyroid hormone, it's going to act as if it wants to be a steroid. This picture is showing you what happens when receptors that are integral receptors found associated with the cell membrane bind their peptide or protein or amino acid derived hormone. Remember, in those they are hydrophilic. I'm not talking about thyroid hormone. They're hydrophilic and the receptors on the cell membrane. Well, then how does the inside of the cell know how to change its physiology? Clearly there needs to be signal transduction. In other words, if the initial signal is stopped on the cell membrane where it binds to its receptor, we have to have a class of second messenger systems. And second messenger systems typically are thought of as cascade-like, or every step you make in this system, it's an amplification, like an avalanche. And I like to think of it as touch tag. You know, I'm it, and then I tag someone else in class. Now both of us are it, and so it becomes 1 to 2 to 4 to 8. Everything's exponential. And that's what happens with every step with second messenger signaling. For you physiology students, you will be thoroughly abused with signal transduction systems, second messenger systems. And in Guyton Hall, they talk about the 3 major kinds, which are shown on the screen. Those of you who are in my pathophysiology, you just need to know that the continuation of that original signal needs to somehow be conveyed deep into the cell, changing the phosphorylation status of current proteins or even changing gene expression. For my anatomy students, you just need to know that if a hormone can't get into the cell, can't target the nucleus directly, we need to have a go-between, and that's the second messenger system. This is going to be my last slide for the first installment, and I want you to see that hormones can be triggered for release in 3 major ways. If we look at the middle picture, let's start there. The middle picture is showing you the neural way. Yes, some axons of neurons, their terminal boutons, or synaptic terminus, can be found embedded in a gland. And really that gland is an extension of the nervous system. So in the picture you're seeing the adrenal medulla. The adrenal medulla versus the adrenal gland. What is that? Adrenal gland means adrenal cortex and adrenal medulla. This picture shows nerves terminating in the adrenal medulla. The adrenal medulla is technically an extension of the nervous system. And when these nerves release their neurotransmitters, they're dumped straight into a bloodstream. And that means that the neurotransmitter is now acting as a chemical signal in the blood, which is technically a hormone. And the best examples of this are epinephrine, which is shown here in the middle picture, oxytocin, and antidiuretic hormone. Some hormones are triggered for release because there's something in the blood, some sort of chemical or compound in the blood that's being monitored. And when that compound or substance gets too low, then hormones are released so that the substance is built back up or accumulates in the blood. If the substance gets too high, then hormones can either be stopped for release or a different class of hormones. Now, the classic example of this would be of a humoral release is glucagon and insulin. If blood glucose levels get too low, then insulin is released. And what does that do? It raises, if blood glucose levels get too low, let me redo that one. If blood glucose levels get too low, glucagon is released. Silly me. Glucagon, its name tells you, glucose is gone. So glucagon will target the liver, and the liver will participate in glycogenolysis. The liver is a primary site where we store glucose in its polymer form. That means, we're attaching monomers together to form a big polymer. So glucagon stimulates the hepatocytes to take glycogen and sever the bonds through hydrolysis in between the glucose molecules and release glucose into the bloodstream. And that raises blood glucose. Conversely, if you have just eaten and your blood glucose levels are rising, insulin tells insulin-dependent cells of the body to insert glucose transporters, and then the cells internalize the glucose from the blood, thereby lowering blood glucose levels. So both glucagon and insulin are examples of humorally released hormones. Humor means of the blood. But they're not the only ones. Parathyroid gland is monitoring the calcium levels of the blood, and when they get too low, parathyroid gland releases parathyroid hormone, and that has 3 major target sites, which I will cover towards the end of this lecture. And the end result is that calcium levels go back up. Just to name a few. Now, the major way to get hormones released is through a hormonal pathway. And typically, when we describe a hormonal pathway, it means one endocrine gland releases a hormone that targets a second endocrine gland that releases a hormone that targets a third endocrine gland that releases its hormone. So we usually say HP blank axis. And the H stands for hypothalamus, and the P stands for pituitary, and this is shown in the far right picture. And X in my slide represents that third downstream gland. There are some notable HP blank pathways. There's the HPT for thyroid, HPA for adrenal gland, HPG for gonad, and a newcomer is HPL for liver. Alright, so that's installment number 1. I'll see you soon, talk to you soon for installment number 2.